I ran into Rich. I don't even remember exactly how. I guess it was through Terry. Terry told me that if you want somebody who knows something about native plant propagation, you should talk to Rich Marovich. And he not only knows about plant propagation, he's been the stream keeper for the Lower Pewter Creek Coordinating Committee since it was formed in 2000. I won't go into the gory details of why it was formed. They've recently completed a weed control and channel realignment project at Winters, and they've initiated invasive weed removal and channel restoration on Lower Pewter Creek and its tributaries, mostly on private agricultural lands. Rich, today, he's going to talk about his native plant nursery at the L.A. Moran Reforestation Center. We'll also have a presentation by Rich in the future on his riparian restoration work. So, Rich, are you on the phone?
Yes.
Okay, well, turn it over to you then.
All righty. Well, thanks, Richard. Well, first of all, it's a delight to be on the agenda, particularly with Terry, who I met in 2008. We are interested in native plant propagation to support restoration projects on a 2,000-acre riparian corridor of Lower Pewter Creek, flows out of Lake Berryessa due east about 30 miles to the Ola Bypass. And we also have restoration projects on some of the major tributaries. The channel of Pewter Creek was degraded by gravel mining, channelization for flood control, and invasive weeds. And we have about 10% invasive weed cover on Pewter Creek, which is not too bad considering that it hadn't been actively managed until about 2000, at least in any organized way. And that means that our native vegetation that remains is very weed resistant and is a healthy restoration or healthy ecosystem. So, but we grow native plants to plant on restoration sites because of the channelization past gravel mining and other disturbances of the channel. A lot of the channel needs to be regraded to be in a natural balance with the current flows, particularly after Berryessa Dam. And so we often need to plant native plants, and when we do, we want to use local ecotypes. And in that, we found common cause with CALFIRE in the preservation and propagation of locally adapted native plants that are adapted to our climate and soils and have a better chance of thriving. On Pewter Creek, our native vegetation diversity is very low because of past disturbances. We have a very active wildlife monitoring program, fish and wildlife monitoring, for over a decade by UC Davis researchers. And we found that wherever we do native plant restoration, we tend to get very rapid wildlife responses. So wildlife respond to the greater diversity of our planted sites compared to the existing prior condition. So I've learned a lot from being at L.A. Moran Reforestation Center, I think by osmosis. I think just being in that facility and seeing how commercial nursery production is done and having Terry to draw upon as a resource for processing and ripening native seeds, picked up a tremendous amount of information. I didn't know very much about propagating native plants when we started, and that's not uncommon. Most people who are doing this that I've talked to have experimented with different methods, and they have succeeded by trial and error, and we're certainly no exception. What I'll be presenting today are the methods that work for us, but as EPA likes to say, your mileage may vary if you try to use our methods, or you may have superior methods, and I would certainly like to hear from you about your experience if you have experience with growing some of these native plants. So who are we talking about? Well, CALFIRE is the manager and operator of the facility. Terry works in the adjacent seed lab. I work in a small portion of the adjacent nursery, about a third maybe of the nursery space right now. And I work for the Lower Poudre Creek Coordinating Committee, which operates like a subdivision of Solano County Water Agency. They're our fiscal agent. And then Poudre Creek Council, which is a public interest advocacy group that organizes community volunteers to grow plants at the nursery and also to install them in our native restoration landscapes. And then the UCD Center for Conservation Biology, who actually got me started propagating native plants with collecting and growing acorns. So the L.A. Moran Reforestation Center was established in 1917. It's home to the only state-run seed bank in California. It's a 40-acre nursery there. Once produced 400,000 conifer seedlings per year, but the nursery operations closed in 2004 due to state budget cuts. We expect that someday the state nursery operations will resume conifer production, but in the meantime, we're diversifying the experience in growing riparian forest species. So not so much the conifers, we grow just a few conifers, but trees and shrubs that are native to our riparian forest. So this is the location of the L.A. Moran Reforestation Center. It's 5800 Childs Road in Davis. It's about a mile east of Mace Boulevard on the Childs Road, which is a frontage road for Highway 80. And then we operate within this red area with a 1,000 square foot glass greenhouse and a quarter-acre shade house and adjacent portion of the potting shed building. We've been operating there since February of 2008 under a MOU with Stephen Jones, who was actually on the cover of the opening slide. Our MOU exists until it is withdrawn by either party, by CALFIRE or by us. We are figuring that we're there on an interim basis until the state nursery operation resumes. But in the meantime, we've learned a tremendous amount, and the knowledge will go with us wherever we continue our nursery operations in the future. So why are we doing this? To meet local demand for indigenous native plants. I should say indigenous locally adapted native plants. We harvest our seeds and cuttings from the Poudre Cache bioregion, which consists of Poudre Creek drainage and the Cache Creek drainage to the north. And we use these plants for publicly supported restoration projects. So our ability to grow plants at the nursery and plant out these plants in our landscapes leverages all of our restoration projects, which are funded almost entirely by grants from the state, with some federal grants and some local funds. All of our work is done by community volunteers. I'm out at the nursery typically every other day to monitor the conditions of the nursery, but then about every other week we have nursery events for community volunteers organized by Poudre Creek Council. They sign up online. We limit the enrollment to 15 volunteers per event so we can keep them well supervised. And they come and they do all of the nursery operations, from seeding and sticking cuttings to transplanting. And then many of these same volunteers will join us for field plantings. So here's a list of trees that we've produced so far. This is a list that tends to grow a bit over time, but this is most of the native species that occur on Poudre Creek. We grow the bigleaf maple, which is currently very limited distribution on Poudre Creek, box elder, which is a little bit more common, alder, white alder, which is fairly common but not as abundant as I think it could be. We have Oregon ash. We actually have a bit of Modesto ash, but that is probably invading from local landscapes. We have some gray pine in the upper part of our watershed. Cottonwood is a very important canopy species for our riparian forest, and because of all these past alterations of the channel, cottonwoods have a difficult time reproducing themselves, and so most of our cottonwood trees are maturing or even senescing, and there is a shortage of naturally produced seedlings to replace those trees as they die off. And natural events such as wildfires have impacted our cottonwood populations. And anytime we have a cottonwood canopy, we find that it's easier to grow all of our other native plants in the partial shade of cottonwoods. So one of the objectives is truly reforestation, to try to reestablish as much of a cottonwood canopy as our current flows will support, and that in turn supports the diversity of other native vegetation in the shade of cottonwoods. We're right on the boundary of the coast live oak. Coast live oak is in Solano County. It's a bit experimental on some of our restoration sites. We have also blue oak, Quercus douglasii, and of course valley oak is our dominant oak on Lower Poudre Creek. We have some live oak, Quercus wislizeni, and it is not very widely distributed on the creek. It's more in the upper watershed than it is in the lower portions. So it's a little bit experimental in our landscapes. We think that we don't have the diversity that was probably once present on Poudre Creek because of the impact of invasive weeds and past vegetation clearing for flood conveyance. So riparian vegetation is much more limited in number of species than one might expect. We have Gooding's willow, which is another canopy species, which we're trying to establish in mixed stands with cottonwoods. And then we have other willows, including red willow, yellow willow, and mostly arroyo willow, arroyo being the predominant species. These are wonderful plants for bird habitat. We have over 220 species of birds that use Poudre Creek at least part of the year. And then we have California sycamore, and California sycamore hybridizes with the London plane tree, so we're always suspect about collecting seeds around cities. So I go north for about an hour to Sycamore Slough, which is a very promising name, and there's some magnificent California sycamores that are well isolated from London plane trees and from cities. They're out in the middle of farmland, and so I feel pretty safe about collecting seed from there and getting the true native sycamore. And then we have California bay trees in the foothills and thinly distributed on the lower part of the creek, and we're trying to increase the distribution of that species as well. Our goal is really to start out with a high mixture of many species in our restoration sites, and then we expect that they will self-select, and some will thrive and others not so much. But we're introducing a little bit, much more diversity actually than what we find as a starting condition. And then we grow these shrubs. We grow California buckeye and mule fat, button willow, Supplanthus. We have some dogwood, one stand of native dogwood. I'm not sure if it's actually Sericea. I think it's the brown twig dogwood that we have. And then we have toyon, we have scrub oak, and lots of California rose, very useful plant that thrives on the floodplains and up on the high and dry banks. And then we have also the elderberry, Mexican elderberry, Mexican blue elderberry, and the image on the right is a 1-gallon elderberry 3 months after planting from seed. So it's capable of growing very rapidly, and this is the kind of firm plug that we like to take out into our restoration sites. And then we grow a variety of native plant plugs. This one specimen is wrapped in plastic. We typically take them out in propagation trays, and we just band a few of these up for more for samples than anything else. But we grow yarrow, which is very drought resistant, and also mugwort, which competes very well with some of our weeds, and Aster chilensis. It actually has the name was recently changed. I'm not sure what the new name is offhand, but this is the Pacific aster, native to Solano County. Carex barbari is a wonderful native sedge that grows on north-facing slopes on Putah Creek, was probably one of the dominant cover plants underneath the shade of oaks. Beautiful stands, very weed resistant, also a very important plant to Native Americans. Carex nudata is the fountain-like sedge that grows on the edges of Putah Creek and other streams in Northern California and Oregon. And it is very useful for stabilizing the edges of banks, and it's one of the most iconic plants of Putah Creek, widely distributed along the edge of the channel. We grow Carex gracilis, which is a slender sedge. It occurs in the Eola bypass, and so we grow those in plugs and to set out in some of our wetter areas. Grindelia camporum is a gum plant. It actually grows so vigorously in the nursery that we're tempted to just direct seed it in the field because it tends to outgrow its containers awfully fast in the nursery. Juncus balticus is Baltic rush, and we propagate that by division, and it's done very well in the nursery. It's a wonderful plant, again, it's a wonderful habitat plant for the edges of the creek channel. Mimulus is monkey flower. We just started growing that last year. It grows so fast in the nursery that we're actually going to delay seeding in the future because the plants are ready to go in the ground within 60 days of seeding. And so one of the things we're learning is instead of starting everything in the fall and spring, some of these plants could be started midsummer and be at a more suitable stage for planting out in the fall. And then Muhlenbergia is deer grass, and it's from seed collected in the Lincoln area, and we don't use very much of that. It's more of a ornamental type grass for the edges of the riparian corridor. And here's a list of trees and shrubs we grow from seed. We have 2 species of maple, maple-related species, big leaf maple and box elder, white alder, toyon, ash, sycamore, oak, rose, elderberry, and California bay laurel. And here's an example of what maple seeds look like as they germinate. We've evolved different propagation styles for different species. For the maples, we harvest the seed in the fall, typically around Thanksgiving. We soak them for 24 hours, and then we put them in Ziploc bags, just the moist seeds with no other material with them. And then in about 45 days, they start to sprout, and when they do, we just see this root initial forming, and then we pluck these out and we stick them individually as they sprout. So it's a bit labor intensive, may not be practical to do on a large scale, but we get a very high success rate of the seeds that we plant this way, and we move them into typically D40 pots, which are the individual plastic pots that fit within carrier trays. White alder we just started growing last year, at least successfully. I've tried to grow it in the past and found that I wasn't harvesting the cones at quite the right time. Terry touched upon the importance of collecting seed at the right time. For white alder, we found that right around New Year's Day, give or take maybe a couple of weeks, is a good time, just when the cones are starting to open and the seed is starting to shatter naturally. And we collect these cones, put them in a paper bag to dry. Last year I tried starting the seed from January through April and found that the later seeding was more successful. I think the warmer temperatures encourage the plants to grow a little bit better, and also we don't have quite the disease problems that we had starting earlier with cooler temperatures. I found out a little bit by accident that if we soak the seeds for 4 days in bulk tea strainers in a bucket of water with a little small fountain pump to recirculate the water, that the seed will actually sprout while it's soaking. And so after, typically 24 hours would be minimum, but after 4 days we get a fair number have actually started to sprout. And then we mix them with a little bit of the moist seed with a little bit of perlite to dry it out and make it spread better, and then we scatter it on top of Staroblock flats and just water it in. So no covering at all because it needs light to germinate. And we found that we had the best germination success by this method. We also found that trying to transplant the seedlings too early, we had a lot of die-offs. So if we waited until the seedlings were about 4 inches tall, when they developed just a little bit of woodiness to the stem, they handled transplanting quite well, and we had very few losses after that. So just in the year's time, we've learned a lot about propagating white alder, and this is typical of our propagating experience. We'll try a lot of different things. We'll try different timing. We'll try different methods. We'll try different containers. And then when we find something that works, all you have to do is succeed one time, and you know how to do it pretty much from that point on. So toyons were a plant that I struggled to grow early on. I couldn't quite figure out how to extract the seed from the little fruits, which are like miniature apples. And it turns out, and there was some misleading information in the literature and all kinds of kind of wives' tales about soaking the seed in strong coffee to help it to germinate, and doing various fermentation treatments of the seed. And I found I tried all these things and they didn't work very well. And so one day I decided, well, I'm just going to try something simple, and I just extracted the seed in a food processor, and I'll show that a little bit later, and sowed them directly into Staroblock flats, and gave them the most minimum treatment, put them on top of a heating pad, and they germinated within 24 hours in high numbers. And ever since that, I've tried to select the simplest methods. We do find that with toyon, because we're planting the seed directly after harvesting the berries, and the berries ripen about this time of year, that we're planting sometimes into an unheated greenhouse when the temperatures are still fairly cold, and sometimes the seedlings will show a little bit of a purple coloration, and that indicates they're being starved for phosphorus because of cold temperatures of the potting media. And so the addition of just a little bit of phosphorus, not very much because it's easy to burn the seed, but just a little bit of superphosphate, a few granules per cell in the Staroblock containers will eliminate this phosphorus stress symptom. And then California sycamore forms these very tight seed balls. We collect these in the late fall. We have to kind of watch carefully because California sycamore seems to drop its seed balls and its leaves kind of all at once, and so it's easy to miss the window of time for collection. But it's typically around Thanksgiving. By January, it's definitely too late. So once we collect the seed balls, we break them up. We pre-soak the seed for 24 hours, and we found that they need darkness to germinate. So don't want to bury the seeds too deeply. They also seem to germinate better in the fall than they do in the winter. So I think they need a little bit warmer conditions for good germination. The seed emerges in 2 to 3 weeks. We also found that we can grow sycamore from cuttings, but collecting the cuttings at the right time is critical to success. When we collected in the winter months, we had practically no success at all, practically no rooting. But if we waited until March, just before bud break, the success was about 50% from cuttings. And I think the difference is the carbohydrates are flowing at that point, the sap is flowing, and that seems to improve the success quite dramatically. So we also grow elderberry and California wild rose. That's elderberry on the left and wild rose on the right. We collect the seed from ripe fruits, and I'll go into the extraction procedure a little bit later. And then we allow the seeds to dry after we've washed off as much of the fruit bits as we can, and we store them in airtight containers in a refrigerator. Then when we get ready to plant, which we do about this time of year, we take the seeds out of the refrigerator, we soak them for 24 hours, and then we chill the seed for 2 to 3 months or until they start to sprout. On the left, you might be able to see some of those elderberry seeds are just starting to sprout, and the rose seeds look very similar to that when they start sprouting. If we wait too long, once they start to sprout, we really can't seed them very well that way because they'll have deformed root systems. So we want to catch them when they're maybe about 5%, no more than 5% sprouted, and just when the roots have just barely emerged. So if we watch them once a week, and it's very important, and Terry taught me this, that we open up the Ziploc bags when they're in the refrigerator once a week, shake the seed around a little bit to get some oxygen in with the seeds, and then seal them back up again. And in that process of checking on them at least once a week, we can catch them at a very early stage of germination, and then we move directly to seeding them. Because we've stored the seed under moist conditions, the seed tends to stick together, and so one of the challenges then is, well, how do we separate this moist seed? And mixing a little bit of dry material, perlite works very well. We can get the seeds to separate, and then we can scatter them more uniformly over the flat. When I first started growing native plants, I was very meticulous about seeding to get just a certain number of seeds per cell. It would typically take me about an hour to seed a flat. And those of you in forestry practice would realize, that that's way too much time when you're trying to do mass production. So thinning the seed with perlite and scattering it, or putting it in a shaker jar and shaking it over the flats, we got a good distribution of seed in a fraction of the time that it would take for meticulous hand seeding. Then we grow these seedlings on for until they get to be about 4 inches tall. 4 inches seems to be a good size for transplanting. And then for the roses, which are relatively slow growing, we thin them to 1 per cell. So we extract the cluster of seedlings out of the seeding flats and then separate them and replant them into the starter block flats. And then for the for the elderberries, they grow so quickly that we take them directly from the seedling flat and put them into 1-gallon containers. They will tolerate, both these species will tolerate crowded roots, and so we can hold them in starter block flats for about a season, and they seem to be just fine with that. But if we pot them up into larger containers, they grow on much faster, and we can have larger specimens to plant out in the field. So oaks are kind of curious in that we used to sprout them in these sprouting trays on the right, very much like this, where we laid out the acorns on top of a little potting mix, and we waited for them to sprout into the potting medium. But then, as you can see on the left, the oaks will grow down and until they touch the bottom of the container, and then the root will actually deform a little bit, and once they touch the bottom of the container, only then will the shoot start to appear. So we can plant sprouted acorns like in the middle picture and put them into gallon containers, and we won't see sprouts for a month because the root is growing and growing until it touches the bottom of the container, and that triggers the shoot to appear. We found that actually I found out this year that we had left some acorns in a paper bag, and they sprouted just as well in a paper bag as they did in our sprouting trays, at least for the valley oak. And so we could keep track of the sprouting of the acorns much more easily if they were just held in a well, a paper bag or in a just a big tray without potting mix. With the potting mix, they would tend to root into the potting mix, and sometimes we wouldn't see that they had rooted, and so we'd end up transplanting them at a later stage of rooting, which wasn't as optimal. So we like to plant oaks when the root is about a quarter of an inch or so long out of the acorn. Different species of oaks seem to germinate at different times, and a batch of acorns will germinate over a period of several months. And so some are quite precocious and they sprout within a week of harvesting. Others might sit for a couple of months before they start sprouting. So we always plant sprouted acorns so we know that they're committed to growing when we put them into the pots. And we plant them directly into deep 1-gallon containers and grow them on, and that's the size that we take out to the field. So California Bay has these little avocado-like fruits that appear in late fall. I was a little bit late collecting this year, but it was a good year for these for the seed production. Only some trees are fruitful, and I don't know why that is, but maybe 1 in 20 or more, only 1 in 20 or so bay trees will actually have fruits, but when they do, they usually have a pretty good crop. So this year we collected some from a few trees in our upper watershed, and we harvested most of them directly from the tree when they were ripe enough that they came off the tree very easily. And some I picked up from the ground as windfalls, and I haven't collected windfalls of bay trees before, so I kept them separate, and we'll see if they do as well as the ones that were harvested directly. The bay trees actually benefit, the seed benefits from being cracked with a nut cracker. And I'd heard about this and tried it for the first time last year, and it does seem to improve the uniformity of germination. I think the seed coat tends to inhibit germination or spread it out over a longer period of time. We find that the bay seeds, when they were uncracked, would germinate over several months. With cracked seed, they seem to germinate maybe over several weeks instead. So once they're cracked, we soak them, and then we drain the seeds and chill for 90 days, and then plant 1 seed per depot. And then in about a year's time, they're large enough to be transplanted out. So Richard, should I pause to answer some questions perhaps? I see we have some questions.
Well, there's only one question that I see from Christy Wagner.
Okay.
You might want to address that.
Sure.
Now, this also is a time for people to catch up if they got differing internet speeds. Christy was asking if you're using a shaker to distribute seed, is there a risk of damaging the seeds?
Well, when the seeds are, if they haven't sprouted roots, or if the roots are very small, they seem to tolerate that pretty well. Again, when we distribute the seeds in the flats, oftentimes if they are sprouting seeds, like in the case of rose or elderberry, if they've required stratification, if they've really sprouted out more than maybe 1/8 of an inch or so, I'll typically put them through a sieve, and I won't bother to plant the ones that are that have longer roots on them that might be damaged in that kind of handling. But it doesn't seem to damage them. And again, the shaker is just a mixture of seed and perlite in a something like a shaker jar that might be used for Parmesan cheese or something at a pizza place, or for a red pepper flakes or something. And we just kind of gently shake them out, and we seem to get plenty of survival. But then there's a lot of redundancy. We tend to seed a little bit on the thick side. We aim to get maybe 5 or 10 seeds per styroblock cell. And if we get 3 to 5 of those that germinate and thrive, that's very successful.
We're getting a couple more questions, but I think we better move on so that you can finish the presentation. We'll queue up those questions for later. Also, I think a couple of the questions may actually be addressed later on in your presentation.
Okay.
So,
I'll move on then. So
Good.
we also propagate from softwood cuttings for mule fat and button willow. This is a great way to increase the number of plants very rapidly. We can usually, well, for these two species, they will typically root in about 2 weeks under intermittent mist from summer cuttings. And we seem to have really good success propagating from plants that are grown in the nursery perhaps because of their superior nutritional status compared to field collected material. But I do make field collections just to make sure that we get good genetic diversity. I collect from different sites. The little picture in the middle of this slide shows an artificial leaf that we use to control the misting. And then we also propagate Pacific aster from softwood cuttings. In this case, a two-node cutting. So the picture on the right with the two diagonal red lines in it indicates where we would cut this stem. And we'd take the lower leaf off, and we would have just that single node below the soil level and one leaf. And it actually grows very fast from just a single leaf cutting. And we can very rapidly increase our number of plants by this method. And it also roots in about 2 weeks under mist in very high percentage, I would say 90% or better. And then for hardwood cuttings, we found that soaking them in water for cottonwoods and willows improves rooting. In fact, they'll actually start to root in the water. They'll start to form root initials. And if we can aerate the water, as we're doing in this tank, we get a higher percentage and more uniformity of root initials on the cuttings. And we tried doing this in the greenhouse and outdoors and compared the effective temperature. And they will definitely form roots faster in warmer temperatures, but I actually prefer to do this outdoors under cooler temperature because it's a more controlled rate of rooting, and it's less likely that the cuttings will get away from us, that they'll start actually going through root elongation and become difficult to handle at that point, because then we would very definitely have concerns about damaging the roots during planting. So we want to plant at the stage when we're just getting root initials, but before root elongation or at a very low percentage of root elongation. And then when we grow on some of the smaller material in the nursery, cottonwoods and willows have to be promptly moved into larger containers. If they get crowded roots, they don't tolerate that very well, and they can actually start to die off, even in the year that they were potted. So they're so fast growing. So one of the most important things we found was the importance of weed control. These are two of our worst weeds, bittercress on the left and oxalis on the right. Any of you who've tried to grow plants in a nursery probably soon discovered that hand weeding is very difficult to keep up with. We do hand weeding on seed flats in the greenhouse, but in the shade house we rely on a granular herbicide, and we just shake this onto the soil surface. We can actually shake it directly over potted acorns, sprouted acorns, planted, maybe the acorns planted a half-inch below the surface of the potting mix. And we can put this kind of herbicide directly over the top. It doesn't seem to bother the acorns, or at least not very much. And it completely eliminates these two weeds. And it's just night and day difference. If any pots that we don't treat with this granular herbicide somehow will have bittercress and oxalis. These are diabolical weeds with exploding seed pods, and so they scatter really small seeds very widely. And so we keep up with our weeds with pre-emergent herbicides, and so we get out ahead of our weed problems, and that's the only way that I found to do it. We've tried different kinds of pots. Initially, we were using recycled pots from a local recycling center, and we were using the kind of round nursery pots that you might find in local nurseries. These round 1-gallon pots are not nearly as space efficient as the square pots. So for example, we could fit 9 1-gallon square pots, a little bit taller pots, in the space that it would take for 6 1-gallon round pots. And then when we go to transport the plants, having them in these square containers, we can put them into square crates, and we can carry 9 trees at a time, as opposed to the individual pots, which which were harder to carry and also to transport. The individual 1-gallon pots are much more likely to fall over in transporting than the square pots in these crates shown on the right. And there are two sizes of these deep pots that we use. We use the 1-gallon, which are which are 9 to a square foot area, and we use a 2-gallon size, which can fit 4 per square foot. So much more efficient use of bench space with the square pots than the round ones. And then we also use these smaller seedling trays, rigid plastic pots that are reusable, and then we use the Styroblock pots shown on the upper right. This is our potting mix for native plants. This was developed by Betty Young from the Golden Gate National Recreational Area. She operates 5 native plant nurseries in that national park, and came up with this mix. And of the public literature that seems to be available on native plant propagation, she's written a great deal of it. So this has been a very successful mix for us. It costs about maybe $42 a cubic yard, so it's pretty reasonable compared to other potting mixes. We buy it in bulk, 40-yard transfer trucks. We transfer it into military trailers, actually. We store it in the trailers, and we bring over the trailers one at a time to the nursery as we need more potting mix. Each trailer holds about 9 cubic yards, so it's an efficient way to handle our potting mix. We ran this potting mix analysis just to mostly to double check on the pH, but also just to verify that our additives were within the ranges that we were looking for. So I was very satisfied with this most recent batch of potting mixes from last year. The pH is 6.6, which is pretty close to ideal, and the other levels of nutrients that are high reflect nutrients that we added. Alkalinity is a big problem for us in Davis. In fact, one of the conditions of being able to use the nursery facility was that we treat the water for alkalinity. And the nursery had been using acetic acid or vinegar and also phosphoric acid as the primary acidifying agents. But we found that sulfuric acid in the form of urea sulfate or N-furic, combination of urea and sulfuric acid, gives us the best reduction in pH, and a gallon plus or less per 20,000 gallons of irrigation water drops the initial pH from 8.1 to 5.7, and because of the alkalinity of the water, the tendency to reduce or to resist reduction in pH, the pH actually climbs after standing for 24 hours to about 6.5, which we figure is ideal. So we have to overshoot in our acidification to counteract the alkalinity and get into a good range of pH. So I'd like to talk briefly about processing seeds. We collect our seeds in 5-gallon buckets, at least for elderberries like this. We put the elderberry clusters over just a regular nursery flat and use that as a destemmer. And just rubbing the clusters on the flat, the berries fall through and it separates the stems, and then we're left with the berries in the bottom of the container. And then we put them in a food processor with a dough blade, and this is a plastic blade that doesn't damage the seeds, and then puree these berries in just enough water to make a slurry, and then rinse the puree through a set of sieves. This is a number 8 sieve on top of a number 12 sieve, so this will catch the berries up on top and let the seeds pass through. And then with a process of washing and decanting, which is basically just floating off the debris, we get pure seed. That's a lot of elderberries. In fact, I haven't collected elderberry seed for a couple of years. We've been living on this one collection. Then we grow toyons and elderberries in these Styroblock flats that I learned about from being at the nursery. These were the same flats that were used for conifer production. We bought our own, a little bit larger size, but very similar to the ones that were previously used for conifer production. And this is a flat of toyons. We pluck these plugs out, and there are actually several seedlings to a plug, and we trim off the roots so that they don't get kinked when we replant them. And then we plant them individually in these trays. One of the things we learned was we don't want to get too far ahead trimming those plugs with multiple seedlings, because they tend to dry out and it affects the survival. So we don't leave a pile of seedlings anymore like we did in this picture. We put them in a dish of water to keep them moist during transplanting. And this is a new crop of toyons coming up. And a group posing with the crop of toyons. This is actually from the days when we ended up occasionally with more than 15 people per event. We now strictly limit the attendance by signing people up on the internet, and we only allow up to 15 to sign up so we can keep them well supervised. This is a few more than 15 in this photo. And then for sedges and some of our other plugs, we direct seed into containers. We don't do this hand seeding this way quite anymore. We use shaker jars because we get a more uniform distribution of seedlings. This is a bit overseeded for sedges. We want ideally about 5 to 10 seeds per cell, and this is probably more like 10 to 20 in this photo. The shaker jar just allows us to make multiple passes over the flat and get a nice uniform distribution of seed. And this is a new crop of sedges and other plugs starting out in the greenhouse. We irrigate with overhead misting and sprinkler nozzles. We use misting nozzles in the greenhouse and sprinklers in the shade house. These are Netafim nozzles. I've been very happy with them. And we have a series of injectors that we use to inject different fertilizers that are incompatible with each other, but that are compatible in dilute solution. And so this is a stack of injectors that we have in the greenhouse. And these are our concentrates. We keep the phosphates and the sulfates and the nitrates separate and inject these individually into our irrigation line. And this is a listing of the fertilizers that we use. We don't put sodium molybdate into our injection system anymore because it is our most expensive ingredient at $30 a pound, but it is, or $30 for the amount that we were using in this batch. We apply a molybdenum fertilizer by foliar spray direct to the plants, and it's much more efficient that way. But our fertilizer costs about a penny a gallon, which is pretty reasonable, we think. So our irrigation system uses booster pumps in the upper left. These are pumps that continuously cycle the water into contact tanks, which are outside of the greenhouse. These are 500-gallon tanks. We have a redundant system. We actually only use one of them at a time, but if the pump goes down or if for any reason we need to work on our irrigation lines, then we can switch over to the other system. So we have pump 1 and 2, and the tanks A and B. This is dilute fertilizer ready to use in the nursery. And then the booster pumps have a valve that detects when our sprinkler lines open up, and so it directs full pressure to the sprinkler lines. When the valve closes, the irrigation valve closes, then the diverter valve goes back into a recycling mode and just keeps the fertilizer mix in constant circulation. We adjust our fertilizers based on leaf tests. This is a typical leaf test. And we follow the data. And so, you know, if it seems like we're short on a particular nutrient, we'll keep adding that nutrient until we get a response in the leaf test. If we actually get lower values in the subsequent leaf test for a given nutrient, that is the definition of a rate-limiting nutrient, because it's stimulating so much growth that we're actually getting lower levels of that nutrient in a subsequent test. So we capture this data and chart it out, and this just shows that we were doing some range finding early on in our fertilizing, and we actually overshot some of the values we were aiming for and then lowered the rates to get back down into the ranges we were looking for. But essentially tracking nutrients over time, you can deduce nutrient demand by looking at the response that you get from one leaf sampling to the next. And we take monthly leaf samples, so over time we can determine if a nutrient is increasing, then we cut it back. If it's decreasing, we add more. And with that simple method, we can fine-tune our fertilizing. So with that, I'll ask if there are any questions.
Wow, that was great. You ended up just on the button. And there are some questions. Mike, do you want to move the questions into the center of the screen? Okay, we already addressed Christy's question about using a shaker, but then Kevin Greer wants to know if you've written a book on what you've been doing. Well, and I guess that's essentially what he's asking. Really, is do you have you you know, some besides this presentation and you know your oral history, really, do you have this information written down somewhere?
Well, not yet, but I'm eager to chat with people who want to know of our successes or have their successes that they can share. We're constantly learning, constantly changing our methods, constantly adjusting our fertilizer rates, constantly monitoring our pH of our water, making fine adjustments to optimize the results. And I think, you know, this was the first year that we really started producing elderberries in large quantity, and having a transplantable 1-gallon plug in 3 months was just the kind of results that we wanted.
Oh, that's interesting. So Mike Taylor-Sau, our moderator, or our host, wants to know if you keep track of seed source plants as genetic material, similar to the way CALFIRE does with conifers.
Well, Mike, all I can say is I've got a lot to learn from Terry, because Terry did an excellent job of describing how to track that kind of information. The short answer is no, I haven't, but I'm very interested in developing a system like that. But mostly we want to diversify our collecting points, so we're collecting from different plants in different years, and we try to collect from a range of plants, and so we try to get a diversity of genetic material within the climatic zone that is adapted to Poudre Creek.
You know, I had a question, actually, I'm going to butt in and ask you, you don't have any California walnut on your list of species for Poudre Creek.
Well, we
none there?
don't. Thank you for the question. California walnut is, well, of course, it is native to California, the black walnut, but it is, we think, mostly native from the Calistoga area and not so much native to Poudre Creek. We think our black walnut and even some pecan has escaped from cultivation, and we find that black walnut is there's kind of an opportunity cost. Oaks and sycamores would grow in the same condition as black walnut in the same setting in the riparian corridor and have much superior wildlife value. And we find that black walnut is weedy enough that we get enough black walnuts on the creek without growing any. So we don't grow any sandbar willow either because it's kind of weedy on Poudre Creek. And I noticed another question, what temperature is chill? And I want to thank you for that question, Herb, because I found out that our refrigerator was set a few degrees too cold last year, and it really interfered with the ripening of the seed. So 42 degrees is the temperature that we aim for, and the difference was our refrigerator accidentally got set for 38 degrees, and the seeds took much longer to ripen, and they didn't ripen nearly as uniformly. So 42 degrees is what we use.
Oh, Rich, do you welcome visitors to your facility?
Well, it's by appointment and subject to CALFIRE policies, but I think during business hours I could certainly meet visitors and give them a tour. And I think anyone who's in the forestry business, you would be welcome and I would just have to do it by appointment, let you into the facility and guide you around and let you back out again.
I think I would like to set up a field trip associated with, the webinar series to some of your restoration sites, and maybe the greenhouse could be included on that.
Sure.
Maybe we could go up to Winters and then come on down and take a look at the growing facility too. So people should keep in touch on and looking at the website, because we will be setting up some field trips to look at some of these case study areas.
Very good.
Here's another question from David Harden. Any seed bank source for the type of species you are raising for the western slope of the Sierra, 2,500 feet on up?
Well, I don't know of any. We collect our own seed. I've been reluctant to purchase seed, even of the same species that we grow, because of uncertainties about elevation and conditions under which they're harvested. So Terry might have a lead on that.
I think one of the questions would be, what species occur at an elevation of 2,500 feet and above? You're likely not to